Good evening. I'm a clinician, so I'm going to just take some examples of some studies where the study was stopped early. Not from us. I just want to take it from a clinician's perspective. Like these are some of my thoughts. So two drugs, and I'll just take 10 minutes. The first drug is remdesivir in COVID patients, and the second one is this abelacimab in patients with atrial fibrillation. Uh, this is the first paper published in November 2020. It was a very famous paper. It's called the ACTT1 study, and it was funded by the NIH. So the patients were those with COVID. The PICO is not cut. The patients are those with COVID. The study drug is, that's okay, no problem. The study drug is remdesivir, and the placebo, it was a placebo controlled study, and some places they didn't have placebo, so they used saline. And the outcome was a clinical ordinal scale. I'll tell you about that. So it's basically patients with moderate to severe COVID. So that's the PICO, so randomized control study. Um, the outcome I told you was an ordinal scale. So what is that? It's not just mortality. This was a scale which WHO came up with. One was very good, and you can go 2, 3, 4, 5, 6, up to 8. 8 is as good as dead. So for some reason, they decided that this will be the outcome of their study, an 8-point scale. Uh, After some time, they decided to change the primary outcome. And they changed the primary outcome to time to response in 29 days. Changed to a comparison of time to recovery. So by day 29, how many people had recovered, and they had defined what recovery was. So they changed the outcome halfway through. This was a strange thing. Another problem with this was the DSMB planned to meet halfway through, but the recruitment was so fast because COVID numbers went up faster than they expected. So the trial finished by the time the DSMB got to meet. So the study stopped. Sorry, the study was completed, and the DSMB met after the all patients had been recruited. So that was an odd situation, and they said they did not expect the numbers to be so high. So this was an ulta situation where the DSMB met after the, instead of interim, it met after the study finished. Now, what happened was the DSMB board members gave the results of the preliminary analysis report and mortality data from the closed safety report was provided to the sponsor, which was the NIH NIAD. The NIH decided that the study should stop. So the sponsor decided based on the report that the study should stop. And since everybody was recruited, they said that no, now what we're going to do is all those who got placebo will immediately be given remdesivir because it's not ethical to give them placebo any longer. So though recruitment had been, they'd already been recruited, those who had got a couple of days of placebo or saline immediately was given remdesivir because it's unethical to continue the study. So that's a plot of the story. Yeah. So this was the outcome, why they stopped. So the DSMB board members highlighted that the median time to recovery was 10 days in the remdesivir group and 15 days in the placebo arm. So the hazard ratio was 1.29, and that was significant. And so the DSMB board they didn't recommend to stop. They just gave the data to the sponsor, and sponsor decided to stop the study. Now, this is where the plot thickens. Mortality over the 29 days was, the hazard ratio was 0.73, the confidence interval 0.52 to 1.03. So it just crossed the significance for mortality. So in the end, NIH decided that the remdesivir will become a drug which must be given to everybody with moderate to severe COVID. And so the practice changed. And there was some politics of that. I'll talk that in a few seconds. So just to say, sometimes this controversies when you get in an article, if you're reading and wondering why did they stop, it'll always sometimes be in editorial or sometimes in these medical journals or commentary journals. So this came from a journal called Stat, and then they talked about this remdesivir and the controversy behind the decision to stop early. So that even in the NEJM editorial, they didn't talk too much about why it was stopped early. But I just wanted to bring out some points from this. So they acknowledge that there's a balance of safety and effectiveness versus as quickly as possible. So this was a pandemic and there was no treatment. So there was some pressure on the government to get some drug because there was no drug till that time. So that was a little bit of pressure from the government. Okay, there's a time to recovery is faster. Let's give the drug and stop the trial. But then they also make a comment at the heart of the decision about when to end the trial was a process that was, as is often the case in clinical trials, by turns secretive and bureaucratic. So this is a group of not doctors but journalists who are science journalists who felt that the process of the DSMB and the sponsor was secretive and bureaucratic. So the NIH, they said they felt that this commentary was it was a fateful decision to start giving remdesivir to patients who had been assigned placebo, limiting the ability to prove mortality. They also picked up the scientific research journalist that there was 8% mortality in one arm, 11% in the other arm, but this did not reach statistical significance. So they were ruining the decision by the NIH. The NIH director got angry because there was criticism about this. So he replied, there was certainly ununanimity within the institute that this was the right thing to do. While I think there might have been some discussion, because everyone always tries to play devil's advocate in these discussions, I think there was a pretty uniform opinion that this was what we should do. So sometimes these decisions are not that simple and straightforward. Even the sponsor had sort of discussion about what to do. What a famous researcher said, I believe in society's best interest to determine whether remdesivir can reduce mortality. And with the release of this information, doing a placebo controlled trial to determine if there is mortality benefit to be very difficult. Because now everybody thinks that I should get remdesivir. So who will agree to be part of a trial to see whether it reduces mortality? So they felt it was a double whammy on society. This guy is the director of the Sloan Kettering Cancer Institute. He said, the reason we have shut the whole society, that is a lockdown, is not to prevent COVID-19 patients from spending a few more days in hospital. It is to prevent patients from dying. Mortality is the right endpoint. So there was severe criticism about the decision to change the endpoint from a ordinal scale, which included death, to suddenly time to event, and then stop the study early and then give everybody. So just to say that this is one example where people were questioning the DSMB and the sponsor. There's no right or wrong decision. There were 1,000 patients in the study, and they recruited all 1,000. But only 480 had finished the 29-day follow-up. Their argument is, why don't you let everybody finish the follow-up and then see what happens? But the researchers stopped the study early because they said that the rest of the 600 patients were being put at a risk, and therefore they were all given, on the placebo arm were also given remdesivir. And they were saying, like, what is the p-value of the preliminary analysis? 0.059. So the NIH said, how many patients would we want to put at risk of dying to get that 0.01 in the p-value? So why are we chasing p-value? So they argued the ulta way. So I'm just saying that you can play every argument in any way. There was an unsaid story, which I picked up in another article, that there was a Chinese RCT on remdesivir. It was going to be published the next month. So there was a big urgency to make sure that they got that trial out first to show that remdesivir worked. In the end, the Chinese trial found it didn't work. And then there was some politics. This was a Gilead study molecule which had failed in Ebola. It had also failed in some other viral outbreaks. So there was a push by Gilead to get it in. And so there was some politics behind it. Just to say that sometimes these discussions are like we had hot discussion with Dr. Khan presented. This can be also slight political decisions about this. So that is the first study. Any questions? Just to show some from a physician's perspective.
The entire recruitment is over. Just theoretically asking, was it within the DSMB's purview to ask for an interim analysis? They anyway finished recruitment. What was the need for an interim analysis then? They could have just.
No, because the follow-up is 29 days. So their argument was that in that 29 days, somebody is getting harmed. I don't think they thought like that. It just happened that the study finished quickly. They just had the data and then they decided, okay, let us stop this study. I don't know. That was there.
No, but the DSMB is there, and there is a need for safety considerations, right? And this was, I mean, I don't know what interim monitoring guideline they had decided on, right? But just because the recruitment has ended doesn't mean that the DSMB board activities end. The board's activities end when the last person followed up ends, not recruitment.
That is just an interesting story. The second one is patients with atrial fibrillation, which is irregular heartbeat. The current standard of care is this newer oral anticoagulant. One of them is called rivaroxaban. And there's a new molecule called abelacimab. This works at a pathway called factor 11, whereas the other original molecule works on this factor 10 and factor 2. So this study was again published in the New England Journal of Medicine 2 months ago. And the main endpoint, these are patients with atrial fibrillation for the patients. The new drug was this abelacimab. The control was rivaroxaban. The outcome here was not, patients with this irregular heartbeat will get strokes because blood clots will happen in their heart and you will get paralysis or some other major blood clot all over the body. So you give these blood thinners to make sure the blood flows properly. The problem with giving blood thinners, you can bleed anywhere. That's a harm. This study did not look at the benefits at all. The major outcome was the harms in the two arms. So this is like the inversion of the kind of a study. So they call it phase 2/3, looking at safety of the drug rather than efficacy. They said another efficacy trial is coming up. So this was, they planned 1,287 patients and they did a randomized active control study and it was in 7 countries. So the statistical analysis was a time to the first event of a major or clinically relevant non-major bleeding. And they had a sample size that said they needed 1,200 patients, followed for a total duration of 27 months. So this is the architecture of the study. They're looking for bleeding, not for preventing of stroke. They call their data monitoring committee as DMC, as Dr. Kant was saying, there are many other words people can use. And they had a DMC charter. I could not find that DMC charter. Do they publish the DMC charter anywhere?
The field files or draft, they need to do that. Like if they have registered into a.
This was a registered trial. The protocol was there, but not the DMC charter.
The protocol has to be registered.
That is there. But
And sometimes the SAT is registered.
Oh, okay.
But the charter, not necessarily.
Okay. Here, so I
If
was.
it's an regulated trial, let's say the FDA trial, they're submitting for indication, they need to.
Submit that. The DSMB, or the DMC in this case, the trial was stopped early on the recommendation of the Independent Data Monitoring Committee because of greater than anticipated reduction in bleeding events with the new drug. So the study was stopped early. So this abelacimab was two doses, 150 and 90. So the major bleeding was 3% in the 150 arm, 2%, 2.6% in the 90 milligram arm, but it was 8% in the comparator arm. So based on this, the study was stopped early. So this was the major thing. So just, I'm finishing now. There were 2 letters to the editor and in the same issue itself. The same, I don't know where this didn't come out in our discussion, so I was curious. So the person who wrote the letter said that rivaroxaban is known to have a high bleeding rate. Even before this study, apixaban is another drug with a lower bleeding rate. So these people have played foul by using a comparator with a high bleeding rate. So you look good because they said the DSMB should have picked up that these people used a bogus, not bogus, but a wrong comparison agent. So that is one criticism.
I agree, but I don't think it's the role of the DSMB. It's the role of the ethics boards, whoever approved the study in the beginning. This is an unethical study to be done.
So this was a question they brought up.
So the DSMB doesn't approve the study. They may see some problems in the protocol or whatever. They're just there to monitor. Somebody has approved it that this is ethical.
That was one question which the author brought up, that this Henry Christopher, that how come this was done in the first place. The second one was this. Another author said, please look at the baseline characteristics. So this Has-Split score is a score to see who has a higher chance of bleeding. And he noticed that the people in the rivaroxaban arm had a higher baseline risk of bleeding. So this is a problem with the interim analysis. Stop early, there'll be differences in baseline. Nobody noticed this. And he further went on to say there were more diabetics in the rivaroxaban arm, more people with heart failure, and more people with prior anticoagulation before this study. So he said this randomization didn't work, or they stopped early. So the baseline characteristics were heavily skewed against the comparison arm. So again, he asked the question like, should the DSMB have not noticed these things and not stopped the study early? I mean, there's no answer to this. The author has replied there was no randomized control study for the first question, apixaban, there's no RCT for this, and some arguments. And the second argument he said was, no, we don't think that these baseline differences are that significant to have caused increased bleeding. So they just put it like that. So I just thought I'll bring these two questions. Sometimes you stop early, it's not so simple as that. It seems to be a complex.
Yeah, but if you look at other things, I mean, you have previous ischemic stroke. There's more in the rivaroxaban than in the others. Previous
bleeding also is more.
Yeah, any history of bleeding is slightly more. Gastrointestinal bleeding more. So they are either randomized. Well, you would not even. There's relatively good numbers. It's still small in comparison to what they could have done. So I'm not sure randomization can be blamed for this. I think there's some sort of bias in assignment that got this like that, because they're starting off worse with bleeding, but in general the rivaroxaban group not only started worse, but it's known to have more bleeding. It should not have been the control arm that they had.
So the question was, should the IRP have told them to continue the study? If you see these results, could we have asked them to continue if we were the DSMB members and we saw this?
No. So before the study would have started, the IRB should have said, this is not the correct thing to do. You know that you're taking a drug that causes a lot of bleeding, so this is not an ethical thing to do, is one. When the study, if they approved it and it comes to the board and the board is constituted, the board members can say, I don't really want to join this board because I think you're doing an unethical study. So individual board members may drop out and say so. Maybe this drug company paid them a lot, who knows, and they decided to be on. But the board members need to think about, is this study ethical, or is the charter something that I want to follow? If I don't trust that, even though they're inviting me, I may choose not to join.
Right.
Yeah. Nobody's forcing you to join the board.
Any other comments? Yeah. So they're arguing that a future trial will look at the benefit. This was a safety part of it. So
I have a question along similar lines. My question: can you test the safety of a drug without proving efficacy?
They're saying phase 2 is the safety, phase 2/3. So
only if it is proven to be efficacious in atrial fibrillation, will it be, or not proven? At least if you have tested the efficacy, can you then go on to say whether it's safe or not? I understand the logic of why they done it. It's more of an ethical question. Like, if you have not tested the efficacy in that condition, how can you jump and test the safety of it?
Well, no, so usually I don't know about Belasimab. Is that a new drug?
Yes, a new drug.
Yeah. So when a new drug comes into the market, there is a whole process of doing trials on it, right? So they've done trials in animals. Then the first use in humans is what's called phase 1, where they see if it's toxic in humans. Then they go to phase 2, and people divide it into phase 2A and phase 2B. Phase 2A is when they're trying to figure out if it is what dose would give some effect. And then phase 2B is to look at efficacy, but they're also looking at safety. And then if it's safe and if it's efficacious in phase 2, then they move on to phase 3 to look at efficacy in a large group. So here, so it's okay to look at safety here in the phase 2B. Yeah, before, because the efficacy will be tested in the big study. So they've designed this one for safety.
But that is in a small group of individuals.
Yeah.
You are testing for. Here they have compared it against another drug which is proven to be efficacious in atrial fibrillation.
Right, and so
I mean, in the sense, how are you maintaining equipoise in that situation?
because they're not testing for safety. I'm sorry, for efficacy. They're testing for safety.
So if you stop it now, in case only you stop the study saying that there's harm for the doctor, you can't then do another, you can't do another trial for efficacy with rivaroxaban at all. Like, see, rivaroxaban is a standard drug, right? Already been practiced.
So their concern is not about the new molecule. Their concern is about this molecule. How will you now do the phase 3 trial if the standard of care is now got a higher bleeding rate? Like, so that's a different
Without
question.
proving that this was actually supposed to be.
Yeah, no, I understand what you're saying, but I'm saying that these people, they're interested in their drug. They wanted to show that
It's safe.
their drug is safe. And they said it causes less bleeding. That's all they cared about.
Suppose I wanted to use a drug which is used for the kidney, like some drugs like dapagliflozin, something which has overlapping benefits on the heart also. I need to first compare it to a drug which is used for the heart and then show that you can use this instead and then test the safety. But these are people who have jumped the fact that you can use abilizumab directly. They've used it for a new condition, but without testing the efficacy and the safety. Like you don't test the safety against the gold standard.
They would have done it like that's
No, but they're not shown it. They're not.
I mean, it's safety and efficacy are two things that you want to test. You can test them concurrently.
But can you test safety before?
You could, yeah.
How would that be? I mean, you can do it. The question is the ethics. If you don't know,
Many people,
how will you then test?
no, but many people argue that safety is the first thing you should be testing. If it's not safe, then why are you going to?
Should you be even there? I mean, not biologically. I understand what you're saying. Point is that you have to first change the gold standard or something, or put safety as a secondary outcome. Here they put it as a primary outcome.
Because that's what they're interested in.
But they've not. Rivaroxaban, you're not. How do you look at it? When you look at a new drug for a condition, you're testing the efficacy of one drug versus the other. You don't test the safety of one drug versus the.
But before you test efficacy, if it's, you want to be assured that it's not toxic, that it's not harmful. So most people do safety first.
But what if it's causing more atrial fibrillation, for example?
Well, then they should have other outcomes that they should be looking at, right?
How do they question the randomization?
No. If randomization work, baseline characteristics should be equal, no?
Not necessarily.
Major confounding factor should be reasonably same.
Suppose I'm doing a study where there is a sex difference because in that population male is more. So when I'm doing sampling, automatically male will represent more. I will be significant.
No, sir. Male will be equal in both arms. Like. I'm saying if there are more men, when you randomize, there will be equal number of men in both arms.
Statistical test and you prove that there are significant difference, number one, and you have to do the randomization, there is a test for randomization, then only you can say that it is not random.
No. Actually, we should not do p-value testing for baseline. We must look and see whether clinically meaningful difference. So whether 3 is clinically different from 2, whether 57 is clinically different from 53. We should not do statistical test. At least that's what I understand.
You cannot say just see the number.
But
By seeing the number,
No, actually, it's not a small number. 430 is a reasonable number.
Big number you are telling, right? It cannot be the value.
No, I'm looking at percentages. 94 versus 90.
Based on that, you cannot tell. That you cannot tell randomization is not. You have to see the numbers equally distributed. That is the importance, not the percentage.
I wouldn't agree.
Equally distributed.
No, that's fine. That's fine.
No, this is different. This,
Whole randomization has to do only with the top line.
yeah.
Do not confuse with imbalances. It's the characteristics of the patient. If
No,
there is
his-
an imbalance.
His,
It has nothing to do with randomization.
I question whether the randomization really worked because the baseline characteristics were not equally distributed.
We have seen scenarios where something like this can happen, but it was being rectified at the early stage because people understood the protocol wrongly sometimes.
And then they,
We end up enrolling patients who are having, let's say we are enrolling a study with Hippocat trial on ejection fraction, what is that? Sorry, the propellant level should be greater than some point. We need to constantly measure to make sure that propellant level is higher than a certain point. And if it is not, it's going to create an imbalance on some of the baseline characteristics like your measurement that you're taking. So we constantly measure it, we constantly see them. But because the randomization is a completely different process that it has nothing to do with the study. The reason is it is independent study. You have a random list and you randomize a patient. And if the balance is not there, of course, there's a problem with the randomization.
that's also the question is, was there a problem with the randomization?
That is only on the first line. That's what I'm saying. 430, 430, 427 tells you that there is a randomization that worked properly.
The baseline.
That means I want to clarify. So there's two things. When you say randomization has been done properly, there's two things that you have to consider. One is, did somebody tamper with the randomization schedule? If to notice that, you would have that the numbers
Are
in
different.
the different groups are substantially different. The numbers in the different groups at the top line are
almost
fairly
the same.
Maybe one, that's because randomness. Now, the balance on the distributions of different characteristics at baseline should be comparable between the three arms. That's not randomization not being done properly. That means you got bad luck. Bad luck in that the people that ended up there were different from the people that ended up in the other ones. Now, how to minimize the opportunity for bad luck is to randomize a lot of people. If you randomize a few people, the chance of bad luck is high. If you randomize a lot of people, the chance of bad luck is small. So when we do studies, we would consider 1,200 or 1,300 people, 1,300 people is a small study. You don't expect that randomization will balance the characteristics, the distribution of characteristics perfectly. But if you look at one of our studies that has 14,000 people, it's very unlikely that bad luck is there. And you would have that the characteristics are balanced. So one thing is the numbers being balanced. The other one is the characteristics being balanced. The numbers being balanced is whether you did the randomization without cheating or what. But the characteristics distributions being balanced depends on bad luck. And usually you have good luck. Most of the times you have good luck. I think they got really bad luck and somebody must have been cheating because you would not have such dramatic differences. If the rivaroxaban people at baseline are much worse off than the two other ones in every characteristic. So that is very suspicious. But I don't know.
Yeah, so that was a second, yeah.
I just had a question related. I mean, we look at our previous exercise in the light of randomization. If we stop a study early, so suppose we are planning to randomize, say, assigned say 10,000 people to whichever group, and we stopped the study because it's showing a significant because of whatever reason we stopped the study early, and we only randomized 4,000 to each, 3,000 to each group. Do we run the risk of seeing when we look at our baseline characteristics that they are not similar because we've still got a lot of more patients to randomize, and is that a consideration also when we are stopping, when we decide to stop?
Right, so you are correct. If you are still recruiting when you happen to have the board decide to recommend stopping the study early, you may get some imbalances if you haven't recruited enough people. But if you had to recruit 10,000 and you're at 4,000, that's a lot of people. So it's unlikely that bad luck is there. But if you are to recruit 10,000 and you're stopping the study after 200 people, then there's likely to be imbalances. And you would, unlikely that somebody would recommend stopping the study that early. There must have been, it could have been for harm because everybody that was in one arm died or something like that. So it's unlikely that you would have a strong effect so early. Now, most of the time, you have to be careful also that most of these long-term studies will take 1 or 2 years to recruit, but their follow-up is for 4 or 5 years. So most of the interim analysis is happening after everybody has been recruited. Most of the time, everybody has been recruited, but not everybody has been followed the amount of time to get. If they are event-driven and you have a high event rate, you may already have enough events with a few people after you've recruited everybody after halfway through the follow-up. If it's number of events driven is usually what we do. Or if it's at a given time, you will have decided when you're going to do it. But most interim analyses are rarely done during recruitment. Now, adaptive designs do their interim analysis. So these Bayesian ones are doing their interim analysis to do sample size re-estimation. And so they then have multi-stage studies. They say we're going to do an interim analysis after the first 1,000 patients to see if we need to get another 1,000 patients, or we only need to get 500 other patients. So they are doing those interim analysis, focusing on optimizing the design of the study, not on necessarily early termination of the study. So the adaptive designs are really sort of a whole other ballgame of trying to do trials. And they have other purposes in doing it. They're trying to optimize costs and study designs and whether we continue or not. And there's other things. Right?
Should they have done an adjusted analysis? This because they had these baseline differences. This hazard ratio, could they have done a Cox model and adjusted for the?
A very good point. That's what, when you have that imbalance in characteristic, especially an imbalance that at baseline shows that these people are bleeding more, I would have adjusted for that analysis.
They should have even talked to earlier that if they're going to expect this kind of imbalance, they should have stratified a randomization
For all those.
Say, randomization problem
that could have been
rectified by stratifying at the early stage.
Look at the reasons why the baseline differences are different, or is it just their role to look at the outcomes? They see more outcomes in the rivaroxaban and they recommend you to stop. To analyze what is the reason behind that and whether the baseline differences were different, that is in a subsequent stage for the investigator to explain.
Yeah.
For the DSMB, isn't it the primary role just to say that the outcomes are worse in this arm? You stop this study in this arm.
Yeah, that's exactly true. It's not their job to do, they're not doing the study. Right?
So the DSMB doesn't look at the baseline and the interim meetings. Do they look at these
No,
tables?
so they would say your primary test is unadjusted under intention to treat, regardless of that, I will look at them. But they also will have seen the
baseline
characteristics of baseline. They said there is residual confounding. I would like to see a Cox model adjusting for that, because I don't think that this is a valid thing. So they can look at it. They don't care why it's happening and that you've designed the study poorly by choosing the wrong that. But I'm now monitoring. I said, well, yeah, this difference is probably due to that. But they may have decided still to stop the study because of you're seeing that rivaroxaban is a whole lot worse than these two. But they say it's probably okay. This is safe. You've shown that it's safe.
Now let's see.
Now you have to report it, but they will have maybe recommended to, when you report this, you should be transparent and say that. They don't care after it stopped. Now it's the New England Journal statistical reviewer should have said, why don't you do
The Cox model.
I want a adjusted analysis here. So they're different people, and it's the reporting of it. They stopped the study because it was, they showed that this was safe. They didn't design the study. They could have said, I don't want to monitor this because I don't think it's an ethical study, but they monitored it. When they're monitored, then they stopped it because they showed the hypothesis.
We would not add the baseline characteristics and take the PI. So how could the PI? I'm talking
Not the
about
PI, the DSMB.
No, I'm saying not about the previous point. You said they could have adjusted if they knew the baseline characteristics.
So once the study is stopped, this is given to everything the PI sees it.
Before that,
PI is not deciding anything, right? So the PI knows the people that they're recruited, and the baseline characteristics by arm are, they won't know it.
So how will they adjust for the group?
No, sometimes, we do. The baseline characteristics is not the outcomes. And so sometimes we will give it to them. They will look at it in the open report.
But doesn't that remove the blindness?
There's baseline values is not a
No,
problem,
not that. I think the study is ongoing.
right?
Recruitment is not done. In one of the interim analysis, you got the baseline result. And you've seen that it's unbalanced. The PI will never know this.
No, the PI knows this. No, we usually don't. No, you won't give them this at the baseline.
How will they adjust for it?
They're not doing any
No,
analysis.
they can't.
They can, they don't do it. But once the study is stopped, then they see it. So when they prepare the paper, they know what they're doing.
If we would have stratified right at the beginning, then it would have been good. But once, so the unblinded statistician and the DSMB can see this, not this particular study because it's all, they have already decided to. But for any other study where things are looking okay, but they see this imbalance, can they then suggest a stratified? No. So you can't go to a stratified randomization after.
Then the balance would have been there.
Right.
But because you know that you're dealing with 400 patients, so there could be an imbalance, and therefore right away think about it at the design stage.
So her question was, can the DSMB now recommend to the PI at the first interim analysis, why don't you guys do stratification?
No,
That was her
you
question.
can't fix
No,
it.
that was her question. Then you're giving away the game.
Now you need to do a adjusted analysis, like that's what.
So her question was, why don't you give it? Generally, why don't you?
No, so DSMB does not advise them on changing the design because of things like that.
The DSMB expressed concern regarding randomization, whether randomization allocation concealment has happened well, if they are suspecting such imbalances, especially one against or in favor of the newer drug, with multiple factors being in favor of the newer drug, when compared to the comparison, whether the DSMB can raise concerns about the randomization and allocation concealment process.
To the
To the
investigator,
To
sponsor.
the sponsor.
To the sponsor.
to the sponsor. No, because you may be then disclosing the baseline characteristics by arm. They may say it in a side way, saying, asking, how is actually the process of randomization being done? Who knows this? Who knows that? They'll ask these questions like that. Could you please clarify how the is it done centrally? Do people call in? Does, who is it done at the site? Is it like looking at envelopes and putting it against the light or what? That sort of thing. They could ask.
Ask for clarification, just a clarification, but I don't think they can actually do anything beyond that point.
Yeah, they can ask
Yeah.
for clarification.
Clarification to understand what is happening, but I don't think they can actually tell something for it to be done in the.
But isn't it technically wrong to continue the study if the DSMB feels the randomization is not being done?
That's why they asked for it.
No, and then so they can, when they recommend to stop the study, on what basis? When they, if this had not shown such a stark difference, if the difference is comparable, and they didn't stop the study, Rivaroxaban is banned for life now, for whatever, significantly the future trial will be affected because this is shown that. If the difference wasn't stark, but the DSMB felt that the randomization was not good.
So here they were not testing that Rivaroxaban was worse.
Yeah, no.
They were testing
Safety of-
that abelacimab was
Safe.
safe. So all their conclusions are it is
Abelacimab.
safe. Whether the comparator, they didn't say anything about the comparator. And it is what it is. And it already, so they're not banning Rivaroxaban. They're just saying, please accept mine. And now I'm going to see if it's efficacious or not.
Yeah, no, but I think if there was no difference, and it does have implications for future trials if you do say this, and the DSMB significantly concerned saying randomization has not been done well, that's why this has happened with this. Can they then recommend that you stop the trial based on that, even if the results didn't show such a stark?
No, but you look at this, the
No,
IQ, the median.
Overlaps.
Yeah, look at the interquartile range. It's 2 to 3, 2 to 3, 2 to 3. There's really no difference. There's just the median is a little bit there. They've rounded it, I think. It may have been, who knows. But look at the percent greater than 3. 52%, 49, or 50, 49. It's not meaningfully different.
Hypothetical situation.
Oh, okay.
That the results are not significantly different. Here there was such a stark safety reason as to why they set the stop.
Okay.
If that was very comparable. But the DSMB feels, safety of the data is also part of their job. They do feel the data collection has not been done well. There's a lot of problems with randomization, there's a lot of baseline characteristic difference. Can they recommend you to stop the trial saying the way you're doing it is wrong and so it's not?
Yeah, they can recommend on operational futility that you are sloppy. This is garbage that what you're going to give me. I don't want to look at your data because I'm not going to trust it because I know that I've asked you for clarifications. I now found out that the way you're doing randomization is by the envelope and people go and look at it. And I know that you're not doing this properly. You're not doing that properly. People are not following. This is going on. That's going on. So this is garbage study. We will stop the study because it's at the end of the day, no one's going to trust your results. And that's futility from an operational standpoint. They can do that. Yep.